Now we're going to talk about the physical foundation, sort of the physics around the biology and the biochemistry we're going to be talking about. So living molecules exist in a dynamic, steady state. They're never at equilibrium. Basically, we always talk about the drive towards equilibrium. If we hit equilibrium, that's kind of like the end. I mean, it'd be kind of like almost like a death. So we need to think about that things are always changing, things are moving around, and a lot of times biology, they're thinking about ways to sort of get around the physics. And it might be taking advantage of it by creating a different step, which then prevents it from being reversible or something like that. So maintaining steady state requires a constant investment of energy. So living things need constant levels of energy. And we'll talk about that a lot throughout the whole semester. So let's go ahead and talk about sort of the thermodynamics and sort of the systems, the physics of what's going on. And the system is all the reactants, all the products, the solvents that contain them, the intermediate atmosphere around it. The universe is the system and all the surroundings around the system. And there's types of systems. There's isolated systems where systems exchanges neither matter nor energy with its surroundings. So then it's kind of isolated. It's not exchanging anything. Closed system exchanges energy, but not matter. So there's energy being transferred, but not matter, not chemistry. An open system, systems exchange with the energy and the matter with its surroundings. So it's a complete exchange of everything. So it's completely open. Everything is dynamic and working in itself. The first law of thermodynamics: in any physical or chemical change, the total amount of energy in the universe remains constant, although the form of that energy may change. So we may have changes in the form of energy, like for example, we can have a buildup of glycogen in the liver, which is sort of saving chemical energy, which can be used again later by doing a little bit of chemistry and releasing that energy. So we have potential energy, it's all the nutrients, everything that's potentially there. Then we have transduction. We can do chemical transformations. We can do heat can be involved, metabolism, breaking things down into making CO2, which you create more entropy, more disorder by making the smaller molecules out of it. And decreased randomness is the building of polymers and things like that is a decrease in the system, but then it allows for an increase in the total amount of energy in the system. So extracting energy from the surroundings, photoautotrophs take light, and you can take CO2 and water, and you can make glucose and release oxygen. And that's kind of classic reaction that we all know of as being something that plants do. So it's light-driven reduction of CO2. So CO2 is in the sort of the optimal, most oxidized form of carbon, and making sugar out of it makes it so it's much more reduced. It's not completely reduced because there's still some oxygen attached to it, but it's getting there. Chemotrophs will take things like glucose with a little bit of oxygen to make CO2 and water, and there is an O2 missing. It's 6 oxygens. It's O2 should be here. It's a typo. So it's energy yielding. So basically, it's the oxidation of glucose makes energy. Autotrophs and heterotrophs participate in global cycles of O2 and CO2 driven by sunlight, making these two groups interdependent. So if you think about it, I mean, some people say that all life is dependent on plants. Well, we get a lot of our food is, ATP, sugar, and things like that are all generated by the fixing, by light hitting and taking CO2 and making glucose. And then there's a series of oxidation reduction reactions, commonly called redox reactions, which you may remember that from maybe Gen Chem or maybe even a little bit of organic chemistry. One reactant is oxidized. It means it loses electrons, and the other one is reduced and it gains electrons. So you can think about oxidation reduction of loss of electrons and the gaining of electrons. So all these reactions are described as sort of the electron flow. And in biochem 2, and in cell physiology and in cell biology, you're going to get to see what some of these reactions are. You see that the biology takes this stepwise version to be able to harvest the maximum amount of energy from these processes. Second law of thermodynamics, randomness in a universe is constantly increasing. That means entropy, which is disorder or randomness in a system, is always increasing. We have to think about biology of making order, making supramolecular structures and all that increases the potential for more and more disorder in a system. So that's why life does not really violate the second law of thermodynamics. Enthalpy is the heat content. It's roughly reflecting the number and the kinds of bonds that are present in molecules. So that's the H in the big equation. We'll be talking about it a lot over time. Free energy of a closed system is the enthalpy minus the temperature times the entropy, which is S. Entropy is S. I don't know why they call entropy S, but that's what they've always done. And so free energy is equal to H minus TS. The free energy change, which is the most common way we see the relationship, is we see the free energy referred to as delta G is equal to delta H minus T delta S. So remember, delta is a symbol for change. So it's a change in free energy is equal to the change in enthalpy minus the temperature times the change in entropy. And S is positive for a reaction that increases the randomness, and H is negative for a reaction that releases heat. And spontaneous reactions all have negative delta G's. So if it's a positive delta G, it's not going to be a spontaneous reaction. Doesn't mean it can't happen, but you might have to do a couple reactions to be able to make it so that it can happen. Coupling reactions. And that's where you have kind of made reference to that. If there's energy needed for a reaction to occur, that any energy requiring reactions are endergonic reactions. And frequently you might couple things to it. Like frequently you'll see ATP, which is this molecule here. You see that those two phosphoanhydride bonds right here have a tremendous amount of energy. We'll talk about that specifically in a future chapter, is how much and why it's there. And by breaking that bond is an exergonic reaction. It releases energy, and that energy can be coupled to a reaction that needs energy to make it happen. So the breakage of a phosphoanhydride bond in ATP is very highly exergonic. So breaking this bond right here is highly exergonic. Coupling reaction. So we think about free energy change of a reaction. Basically work being done is like this thing going up an incline, and there's a loss of energy. It would be the spontaneous reaction, so there's maybe something that pulls it up the hill. So endergonic reactions, this is the energy requiring reactions, and these are reactions that are releasing the amount of energy. And a classic example of sort of coupled reactions, you have the reaction of glucose reacting with glucose and phosphate reacting to make glucose 6-phosphate. That requires a fair amount of energy. Well, if you think about it now, you look at the amount of energy required, the amount of energy that's released by the hydrolysis of that one phosphoanhydride bonds. We have this big decrease of energy. So if you combine these two reactions together, you wind up with one reaction that is spontaneous. It has a negative delta G. So you have ATP being consumed, providing the energy so that that glucose molecule gets phosphorylated. So we basically in biology, a lot of other coupled systems, if you're trying to do chemical reactions, you synthesis of other chemicals, frequently you wind up coupling multiple reactions together so that there's a transfer of energy and you can take advantage of that energy. There's an equilibrium constant, which is the K equilibrium, that's KEQ, and delta G are ways to measure reactions tendency to be able to proceed spontaneous. Let's look at the reaction of A plus B yields C plus D. So the little numbers refer to stoichiometry, and if you go ahead and write this reaction, KEQ will be equal to this equation here. So KEQ is the reactants, the products over the reactants. So these are all the products, and we have, it's always to the power of the stoichiometric amount. If they're just, if it's unimolecular, if it's one of each one of them, it's always to 1, so it's just that concentration. If it's 2 of them, in some cases, they'll be squared and things like that. So keep in mind that this is the way, sort of the way a reaction will occur, and gradually work towards going towards equilibrium. So the ratio of these products over these reactants is driven by this KEQ. So delta G is the actual free energy change of any chemical reaction. So it's the amount of change going from the beginning to the end of the reaction. And you can combine the two equations together, and you'll get delta G is equal to delta G 0 which is sort of the total rate energy of the reaction, standard free energy reaction change, times R, which is the universal gas constant, T is the temperature, and it's the log of that KEQ. So A sub 1 is the initial concentration of A and so forth, and R is the gas constant, and T is the absolute temperature. So we can have this is that general reaction of how much energy in the free change, the change of energy in that reaction occurring. So at equilibrium, G is equal to 0, delta G is equal to 0, and these two things, this is the initial and this is the equilibrium amount. So we can rearrange the equation and wind up with delta G is equal to delta G standard, RT log of this. So it can be delta G, and this is the KEQ. So it's the negative log of the KEQ. And looking at it's the same example they gave you a little bit earlier. Is a reaction 1 is endergonic. It requires energy, so it's got that positive delta G. The second reaction has a really strong negative delta G, so we know that that's a reaction that can occur, and there's a huge release of energy there. And reaction 3 is negative. It's the combination of these two being coupled together. You wind up with it being potentially spontaneous by adding these reactions together. So enzymes promote sequences of chemical reactions. What enzymes will do, and we'll talk about this in much more detail later in the semester, is there's a certain barrier to all reactions. It's referred to as the activation barrier, or I commonly refer to it as the energy of activation. So in a normal reaction without an enzyme present, it'll follow this blue line. So there's quite a bit of energy they needed to do that. What an enzyme will do is the catalyzed version of this reaction will be lower, which means that increases the probability that that reaction will occur. That probability increase means it effectively increases the rate of the reaction. But what enzymes do is they decrease that activation barrier, make it require less energy to get to that activation state, that transition state. All this goes, and as we go in, and this is stuff that you may have had before. It's kind of giving you the precursors to some of the stuff we're going into more detail and talk about how it works and how it's done. So remember, this is sort of this part of the class is sort of a precursor to what we're going to talk about for the rest of the semester. So understanding these basic principles are important. So pathways. In biochemistry, we have these series of pathways that have these steps of making a particular amino acid, or making glycogen, or making cellulose. There'll be a pathway to do that. Now, and if you're going to break something down, you might take, glycogen and make glucose out of it to be able to make ATP out of it, for example, is a common pathway that people know about. So that would be a catabolic, or that would be an example of catabolism. So they'll be breaking down larger compounds, which will drive the manufacture of ATP and produces reduced electron carriers like NAD, NADPH, things like that. Anabolism is synthetic pathways that require the input of energy. So it's making these things. So making complex molecules, building glycogen from the excess of ATP and glucose present in the cell. So the cell always is balancing between these two things to play around with, to be able to get that constant amount of energy required to maintain itself. Metabolism is the overall network of enzyme catalyzed pathways. It includes both the ones that are breaking down molecules and the ones that are building molecules. So it's both catabolism, anabolism. And the unity of life is all these basic pathways generally occur the same way in all living organisms. They might be regulated differently, and they might be used in slightly different ways depending on the environment, how that organism lives, but these reactions generally occur in the same pathways across forms. So we can elucidate pathways in lower organisms to be able to understand the more complex things that happen in humans, for example. We can see the basic pathways, know the pathways, and then look at how they're regulated within a higher organism, a more complex organism. Metabolism is regulated to achieve things. So, we can't have all these pathways going all the time, going all these different directions. That doesn't make sort of energy sense. Because you think, even though animals and all living things are consuming energy constantly, you need to be able to sort of save it and maintain it and not, build up a lot of this one molecule whenever we need a different molecule, for example. So we need to have regulatory pathways. And there's one of the most simple ways is feedback inhibition. And a classic example is threonine is the amino acid that is used to be able to make isoleucine. We'll know the structures of those later, and this pathway, you'll learn about that pathway in biochem too. But what happens is that first enzyme on the path to making isoleucine itself can bind to that enzyme and slow it down. So if you have a lot of isoleucine present, it'll feed back and inhibit that enzyme 1 on the pathway where it will no longer make isoleucine. So when the isoleucine concentration gets too low and the cell needs isoleucine, it will no longer be inhibiting that enzyme and it'll start making it again. So systematic, so it can actually be a little balanced system. And in reality, there are people studying systems biology now that are trying to understand how all these different pathways all interact. If we make this change in the system, what are the global changes? What are all the changes? And those are the people that study systems biology. So now we've talked about sort of the physics. Now let's talk about the genetic foundations. Everyone likes to talk about the genetics about things. I got that from my father, or I got that from my mother, or whatever, or my grandmother, or something like that. So there's genetic foundations. Let's talk a little about that sort of genetics that's relevant to biology. Okay, we're going to talk about it in great detail of how it does it, but we're going to talk about what it does right now. So DNA, deoxyribonucleic acid, is a sequence of monomeric subunits, deoxyribonucleotides, that create sort of a library and an instruction set to be able to make all the molecules that a living organism needs. So it provides the template to produce not only all the proteins and all the RNAs that are needed, but it also provides a template to reproduce itself. And whenever we talk about the structure of DNA in the class, we will talk about the observations that Watson and Crick made whenever they sort of empirically determined what the structure of DNA was. So DNA is this information molecule, and in reality, it's a molecule that has sort of two functions. It basically has a function to be able to template to make everything the cell needs, and it's a template to reproduce that organism. So this is sort of the two things that it needs to do. So its only job is sort of to be that library and provide reference materials. So genetic continuity is vested in a single DNA molecule. So a DNA molecule on the E. coli cell is a single molecule containing about 4.6 million nucleotide pairs. So DNA, what we'll find is a double helix. There's two strands that intertwine with each other, and they need to be replicated perfectly to give rise to identical progeny by cell division. So E. coli has been a heavily studied system, and it has about 4.6 million nucleotide pairs. And here's the little molecule. So you see there's two strands, or strand 1 and 2. They're interchanged with each other. You see that A's are across, A's bind to T's and G's bind to C's, and that determines all that is the information that's stored. It's all stored in 4 bases, and there's a code that sort of organizes all of life together. And these strands are held together by hydrogen bonds, which we'll talk about in the next chapter. So we have the native conformation. For example, in this part of the DNA, there'll be a gene for an enzyme called hexokinase. Hexokinase gene will be transcribed by some proteins to make a messenger RNA, which is complementary to this DNA molecule. And it'll be translated. Translating meaning it takes the RNA sequence and converts that into a protein sequence, which will convert, basically makes this chain of amino acids. That chain of amino acids will fold in a way such that that hexokinase will go ahead and take ATP and glucose, make ADP and glucose 6-phosphate. And you'll see that's the system. So this is also referred to as the central dogma. DNA makes RNA, RNA makes protein, and protein folds and makes the enzymes that we need. Any structural molecule, any enzymatic molecule we need. A little bit about the DNA. We'll talk a lot about that in a later chapter. There's 2 chapters on this sort of nucleic acids and then sort of the genetic techniques that are all based on being the simplicity of the DNA sequence and stuff. Now, let's talk about evolutionary foundations. How did it all come about? We're talking about the constancy of life being all interrelated. So let's go ahead and look at that. So let's look at the evolutionary foundations of life. Let's go ahead and changes in heredity and instructions allow for evolution. For example, mutation. Let's say this hexokinase gene has some error in it. It'll make a different mRNA, which makes a different protein, which may change the activity of that enzyme. So some changes are beneficial, some changes are not beneficial. But that's the key aspect of what is the mechanism of evolution. There are other factors that create and select for the mutations that occur, but it all happens at the level of genes in DNA sequence. So biomolecules are thought to initially be formed. There's these vents, high temperature vents in the ocean, and there's living things there that may have a very, very unusual chemistry. It's part of this archaea that lives there. They live on basically being able to reduce sulfate into sulfide. And Miller and Urey basically came up with ideas that, hey, maybe we could make biomolecules by creating this little experiment they did where they have some of the basic gases that would have been in the early Earth's atmosphere, add a little bit of electricity, from basically lightning, and see if you can make smaller molecules. And they found that they can make a lot of different initial molecules could be made in that way. And so a lot of people think that, hey, it could possibly happen that way. So ribonucleic acid, this thing right now, I think I told you that generally proteins and enzymes do all the catalysis. In reality, in some cases, RNA can have significant biological reactions as well. And it's thought that maybe at one point, life may have been sort of an RNA world where RNA is the central molecule. It did basically almost everything, and then gradually evolved proteins, and then storing that information in DNA came about. And so this RNA is not a stable molecule as DNA. So there's a lot of thoughts about how these things could have evolved and changed over time. Of course, we can't really tell how these things actually happened. So RNA were created precursors, may have been the first genes in catalysis. And you can see a little prebiotic organism, short RNAs. RNAs have catalytic activity. You can make specific peptides catalyzed by RNA. And you can keep doing it. This keeps going on. And it gradually gets to the world that we know now, the DNA genome translated on RNA protein complexes with RNA and protein catalysts. So you can see that there's all these different versions. There are viruses now that are RNA based and things like that. So there's some evidence that these things are happening. So evolution began more than 3.5 billion years ago, and sort of formation of Earth, and we go to here, 4.5 million years ago, and this is sort of the time scale. It took about 3 million years for bacteria, about 2.5 million years for the appearance of aerobic bacteria. Some bacteria maybe occurred after about 1 million years, and then about 1.5 million years ago, eukaryotes first came about. And then into symbionts about maybe 1 million years ago, and red and green algae occurred, and diversification about 500 million years ago. So here's the thought process of how long it took. I mean, it took a huge amount of time to work through all these details. Earliest cells probably took energy from inorganic fuels like ferrous sulfide and ferrous carbonate. Photosynthetic processes arose from evolution. Pigments that capture energy from light in the sun reduce CO2 and organic compounds. Atmosphere became richer in O2 with the right, and then the initial atmospheres were always thought not to have very much oxygen at all. But these organisms start producing oxygen. They get so successful, they actually replace a lot of the CO2 in the atmosphere and methane in the atmosphere with oxygen, and that created an environment for other organisms to grow and evolve. So eukaryotic cells evolved from simpler precursors. There's a big thought. I made a comment about the endosymbiont theory. There's endosymbionts that it's really thought that because mitochondria and chloroplasts all have some DNA in them. So it's thought that maybe a bacteria-like organism invaded inside of a eukaryotic cell and lived symbiotically with that cell and became these organelles. So it's thought that the cell was there and then these things came in. And you have these, so that mitochondria were a bacteria that infected, in plants they have a photosynthetic unicellular organism invaded those cells and then made it so that those cells could produce sugars from CO2 and light. So things keep working out. And here's sort of a system where it's just kind of a description of it. You have an ancestral anaerobic eukaryote that has a nucleus, it has all these endomembrane system and all that sort of thing. And then the bacteria goes ahead and infects inside. Instead of the cell going ahead and degrading that organism, it went ahead and lived symbiotically within that organism. The organism passed food to it, passed things to it, and then it passed things to it. So it became a nice symbiotic relationship. And you developed into a modern aerobic eukaryote. Later on, plants were evolved from having cyanobacteria infect these cells that already had mitochondria and making a photosynthetic eukaryote. So they've thought about how these things could have occurred, and it's kind of interesting things have happened. Let's talk about a little bit about some of the terminology. We'll be using some of these terms to show relationships and other things. Homologs are proteins that are encoded by genes that share readily detectable sequence similarities. It's thought that organisms probably evolved maybe having duplication events, and then one gene gets modified at a different rate or a different way, and it adds some selective advantage for an organism. And so homologs, I'll refer to homologous or similar proteins, and that's where this comes from. So they're encoded by genes that share relationships. The sequences are somewhat similar, but they're not identical. And they can basically determine phylogenetic relationships. So we can kind of get the organisms, and they evolve over time, and all these changes occur over time. And you can see relationships by distance and changes by how long they might have happened and things like that. You can find relationships. So functional genomics shows allocations of genes to specific thought processes. We talked about the constancy of life, that always basic reactions occur. So there's these proteins and genes for specific processes that might be related to other things and evolve and give that specificity to the cell. But then there's this group of other proteins and genes. They're called housekeeping genes. They're expressed under all conditions. They're not subject to much regulation, but they keep that cell going all the time. So it's the way to keep that cell alive. And there's a group of things that can be, during certain situations, are expressed. Genomic comparisons have increased importance in medicine. Large-scale sequencing studies have shown many genes and many mutations correlate with medical conditions. You're hearing almost every day that someone finds that they have this genetic mutation. In some cases, people are doing whole genome sequences themselves to look for some of these things to see what might have happened to them. Maybe they could work to prevent diseases and genetic disorders from happening. So looking at all these proteins. The first human genome that was sequenced was probably several $1 billion. Now you can routinely get your genome sequenced for $1,000. In some cases, I think I saw recently it was done for about $300. So it's getting to the point where maybe having your genetic background might really be able to help you out. But of course, there's going to be privacy issues and all these sort of things that we won't get into in this class. But it could potentially help one medically. And that's kind of the end of this chapter. This is sort of all this biology. These are all things that you probably had before, maybe not quite in this much detail because it wasn't biased towards biochemistry, but hopefully you'll be able to understand these things and work through them. And if you have any questions about these things, feel free to come by and talk to me. We can work through some of these things and give you a background in them. This will get you, this video is meant to just give you a jump start and to be able to understand some of the other things. And we're going to start off start off in chapter 2 instead of chapter 1 in the book. If you have any questions, definitely get hold of me, and we'll see you later. Bye.